Do you use the SIBO Breath Test? Can you identify an insect by the buzz? Can you identify bacteria by the breath test or just their presence? Some bacteria are beneficial. Some bacteria are harmful. Some bacteria kill beneficial bacteria.
And bald-faced hornets attack and kill honeybees. So,
is it hornets or honeybees? Is it good bacteria or pathogenic bacteria? The SIBO breath test cannot identify specific species of bacteria. For example, here you see the results of a SIBO breath test and the Genova GIFX for the same patient. The results for the SIBO breath test are negative, but the GIFX test shows several species in the high 5th quintile range. This leaves the impression that it must not be a bacterial problem if the SIBO breath test is the only test that is used. However, the patient is still having symptoms. With the SIBO breath test, there is no way to identify the bacterial species, and if there's no way to identify the bacterial species, there's no way that we can identify their behavior and how they are causing or contributing to the patient's symptoms. The confusion and differences of opinion in stool testing may be due to the changes made in what is analyzed in the stool test. The elimination of PCR testing virtually eliminating the finding of H. pylori. Fortunately, DRG Labs is now doing PCR testing to replace what Genova eliminated. As you can see in these test results from the same stool sample, H. pylori IgA testing is negative, while PCR testing from the DRG Labs is positive. How can that be? The answer is really quite simple. H. pylori cleave immunoglobulins and also have the ability to hack into the immune system, taking over control of IgA1 and IgA2, and using the IgA for their protection from the immune system. This is discussed in the Mastering the Gut program. SIBO, like leaky gut, is a generic diagnosis. What level of permeability does the patient have? As you will see later in this video, the behavior of the microbes may play a significant role. If the species are determined, their behavior and effects can be correlated to the patient's condition. The Genova GIFX 2200 and the DRG Labs identifies the species and their numbers, something a SIBO breath test cannot do. Here's an example of bacteria hacking into the immune system. The misshapen red blood cell is infested with mycoplasma, another bacteria that Genova eliminated from their stool test. You can see that the macrophages, 2 of them, are attracted to the infected red blood cell. However, they are repulsed and move away swiftly. In addition to the mycoplasma infected cell, there are also other misshapen red blood cells. These are incapable of carrying oxygen to the body. The markers used to identify these cells in CBC lab test is covered in the red blood cell morphology portion of the Mastering the Gut program. You can access this program at masteringthegut.com. In a ghetto gut, gang members, or in this case, gastrointestinal bacteria, shift the environment, making good people, or bacteria, modify their behavior just to be able to survive. The bacterial gang members control the behavior of others. Bacteria hack into the NEI supersystem using hormones and neurotransmitters and the immune system for their benefit. They mimic other bacteria. They cleave immunoglobulins and use them for their own selfish protection. What happens if the biofilm is broken up before getting control of the gastrointestinal environment? The bacteria were contained in the biofilm. Where are they going to go? How will they react? They will seek out a new place to colonize. Can you guarantee where that will be? Because PCR testing gives a much more definite answer on how many colony-forming units are present, as in this case, you can see it's definitely not SIBO. This GIFX result may seem a more likely diagnosis of SIBO with so many in the 5th quintile. However, using the Beer analysis criteria, we must now look at the behavior of each microbial species. What are their effects on the gastrointestinal lining and environment as a result of their behavior? Are there expectations of symptoms they may cause or contribute to? In addition to, what are the test results? This is all covered in the Mastering the Gut program when we're analyzing the gastrointestinal environment. Microbial species have behaviors specific to them, causing signs and symptoms identified in the gastrointestinal assessment forms designed for the Mastering the Gut program. Microbial species also have preferred niches to inhabit in the gastrointestinal tract. It would be inappropriate to make a blanket statement that anything that lands on a flower is a bee, because a wasp lands on flowers and both will sting in self-defense. Microbes have a preferred environment to colonize, just as penguins have a preferred environment. While Lake Michigan in the dead of winter may mimic the penguins' niche environment, they are not found there. The environment would have to drastically shift to support them in the Great Lakes. In this test, with a large amount of 5th quintile bacteria, can you identify the bacteria that behave like moles? Can you identify the bacteria that exhibit termite-like behavior, chewing up the mucosal lining? Can you identify the bacteria found predominantly in the mouth, having survived a trip through the digestive chemistry? Can these behaviors, effects, and expectations be identified using the results from a SIBO breath test? It is highly unlikely. The Mastering the Gut program will provide you with the knowledge and the analysis guides to analyze the results of the gastrointestinal stool test. For more information, go to masteringthegut.com.